Now moving towards the junctional epithelium. So junctional epithelium is a collar-like band of stratified squamous and non-keratinizing epithelium. In this, they have told you about that how it is. It is a collar-like band. Which cells are these? They are stratified squamous, and then they have also tell you that epithelium is keratinized or not. It is a non-keratinizing epithelium. Okay. Then you have 3 to 4 layers thick in early life, but it increases with the years, with the age. Okay. Then it differs from its coronal end and may have the 10 to 29 cells white. It is 10 to 29 cells white and 1 to 2 cells at its apical termination. It is located, very important, at the cemento-enamel junction in healthy tissues. Means it is located in the at the cemento-enamel junction in healthy tissues. Okay. And these cells can be grouped into two strata. They have two strata basically. Okay. They have two strata. Basically you have, like, they have grouped them in two strata. Either the basal layer that faces the connective tissue. They have two layers. Or either suprabasal that extends to the tooth surface. So they have two layers, junction epithelium. One layer faces to the connective tissue and the other layer is faces the tooth. The length of junction epithelium ranges from 0.25 to 1.35 millimeter. The junctional epithelium has also influenced by the oral epithelium. Well, it is formed by the influence of the oral epithelium and the reduced enamel epithelium during the tooth eruption. However, the reduced enamel epithelium, it is not important for its formation. In fact, the junctional epithelium, it is completely restored after pocket instrumentation or surgery, or it forms around an implant. Means it is not basically the, you can say, formed by reduced enamel epithelium. Yeah, it is like, it is like, if for its formation reduced enamel epithelium is not important. It basically totally restores it itself. Okay. So now basically it is attached to the tooth surface, to the tooth surface, that by epithelium attachment by means of interdental basal lamina. Okay. It is attached to the tooth. It means it gives a support. It is also attached to the gingival connective tissue by external basal lamina. It has the same structure as the epithelium connective tissue attachments elsewhere. Means one side is like internal is connected to the tooth and the external basal lamina is connected to connective tissue. Okay. Then internal basal lamina basically consists of the lamina densa. Lamina densa is basically internal basal lamina consists of lamina densa basically, or lamina lucida, to which hemodesmosomes are attached. So basically this densa layer is towards the connective tissue and lucida will be towards the epithelium. Always remember, hemodesmosomes have a decisive role in the firm attachment. Means they have a firm attachment to the of the cells to the internal basal lamina. They have a firm attachment role. Okay. Data suggests that these hemodesmosomes also has a specific size for signal transduction. Means they have a specific size of signal transduction. Okay. Signal transduction, and maybe participate in the regulation of gene. Okay, very important. They can also participate in the gene expression, cell proliferation, cell proliferation. Okay. So they have also the cell proliferation role, very important. They have cell differentiation role, and organic strands from the enamel appears to be extended into the denser. Junctional epithelium attaches to afibrillar cementum that is present on the crown. Okay, afibrillar. Okay, basically usually it is restricted to the area within 1 millimeter of cemento-enamel junction and root cementum in the similar manner. And then the histochemical evidence also shows the presence for the neutral polysaccharides in the zones of epithelium attachment has been reported. It has also polysaccharides. Data shows that the basal lamina of the junctional epithelium resembles to the endothelial and epithelium cells in its lamina content. Means they're saying that the basal lamina is present in the junctional epithelium similar to the endothelium and epithelium cells in the laminin content, but it differs in the interbasal lamina which has no type 4 collagen. These findings suggest that the cells of junctional epithelium also has the production for laminin and play a vital role again. Adhesion mechanism, very important. Lot of things we have said now. Then attachment of the junctional epithelium, they say the attachment of the junctional epithelium to the tooth is reinforced by junctional fibers. Of course that the junctional epithelium is connected to the tooth by gingival fibers, which basically embarrasses the margin gingiva against the tooth surface. For this reason the junctional epithelium and the fibers are considered together a unit, which is gingival dento-gingival unit. What is dento-gingival unit? When you combine the junctional epithelium and gingival fibers together as a unit, it is dento-gingival unit. In conclusion, we can say that the except that the gingival epithelium has shows several unique structure and functional features, it also contribute to the prevention of the pathogenic bacteria flora from, it does not colonize the bacteria from the subgingival water surface. So it is, so first the gingival epithelium is firmly attached it, basically it is firmly attached it, so it is firmly attached to the tooth surface, number one. Then due to which it forms a barrier, okay, barrier against the plaque. Second, it also access the gingival fluid. It has inflammatory cells and components for immunologic host defense to the margin gingiva. The third, it shows a rapid turnover which contributes to the host parasite equilibrium, means it has a rapid turnover time. Okay, then the rapid taken due to the rapid repair of damaged tissues. Some investigators have also indicated that the cells of the junctional epithelium has the endocytic capacity equal to that of the it has also endocytosis like macrophages and neutrophils, that is the activity may be protective in nature. So it has a lot of function. It is basically a barrier, it is basically, it has endocytic cavity, basically it should has a rapid turnover rate, means cell proliferation. It has a lot of functions. Prevent the entry of pathogenic bacteria. Thank you.